So our next speaker is Jerry Leung, who will speak to us about the correlation between platelet donor characteristics and quality metrics to platelet transgenic protein production. So Jerry, thank you very much for speaking.
Thank you for the introduction. My name is Jerry Leung, and as mentioned, I'm a postdoctoral fellow co-supervised by Drs. Peter Cullis at the University of British Columbia and by Dr. Christian Kastrup at the Diversity Blood Research Institute in Milwaukee, Wisconsin. And happy to talk about some of our work that we did to try to look between a correlation in platelet donor characteristics and platelet transgenic protein production. And just waiting for the slide to move over. Yeah, so just to give a brief introduction, and I'm sure all of you here are already aware of the importance of platelets during transfusion and in their role in participating in injuries and hemorrhage, where during an active bleed, for example, platelets become activated and form a platelet plug that helps to facilitate hemostasis. But one thing I'd like to highlight is that a lot of this process in terms of platelet activation is driven by changes in the platelet proteome, whereby you have a lot of proteins being highly expressed during this stage, and other proteins that are downregulated, also largely driven by effects and changes in the microRNA of a platelet, indicating that platelets are capable of protein expression during different stages of activation. One thing I'd also like to highlight here is that platelets also, during storage, for example, are capable of also expressing different proteins, and is highlighted by this graph here, demonstrating an increase in glycoprotein 3A during storage, again, indicating that platelets themselves have some kind of translational capacity to translate different proteins, either under storage or activation. But one big question remains is that while we do understand a lot about what happens with platelets when they become activated and during hemostasis, there's much less known about the actual translational capacity in platelets and actually what are the factors that govern protein synthesis in platelets to begin with. And so to try to begin to answer this question, we relied on some technology that we recently developed in the lab, whereby we were able to take lipid nanoparticles encapsulating messenger RNA to deliver these into platelets in an ex vivo manner and express exogenous proteins. And so what we have been able to achieve recently was to take these lipid nanoparticles and express either a firefly luciferase reporter or a nanoluciferase reporter in platelets that are either stored in a Tyrod's buffer, which is typically used for washed platelets, or those that are a little more clinically relevant in a PAS 7030, so platelet additive solution supplemented with 30% plasma, or just in plasma alone, where in all cases we're able to see the expression of our nanoluciferase reporter in these platelets. And so using this technology and having the opportunity initiated by Ellen with her work that's described in her previous talk, we were able to try to take these lipid nanoparticles and look at the translational capacity and correlate that across different donors. And so again, having the opportunity to access a large pool of donor platelets, we then transfected these with our lipid nanoparticles and then looked at the expression of nanoluciferase and then characterizing these platelets and donors at the same time to see whether or not there's a correlation. And so one thing off the bat, unfortunately I don't have any good news to say that there is a positive correlation because we really didn't see any correlation at all, which may not necessarily be too surprising. So one thing we did note was that across all 81 donors that we did test, we were able to see some degree of nanoluciferase expression in our platelets. However, when we start to look into the different kind of metrics that we're measuring, for example, the donor age or the blood type, the donor sex, the diabetic status, alcohol consumption, or the tobacco smoking status, we saw no correlation at all. That was also statistically significant. And you can see it's highlighted by the wide variability in expression that is spread out across all these different metrics. When we want to look further into the platelets as well, because those were all the donor characteristics, and looking into the platelet characteristics themselves, and here's just a sample of some of the things that we measured, specifically the activation status of platelets, because again, platelets are known to express proteins during activation. We also did not see any kind of correlation. So again, highlighted by, for example, in the graph A here, that across platelets with some kind of baseline level of CD62P expression, or when you treat them with a mild agonist, such as in panel B with ADP, we do not see any kind of correlation between the levels of their degree of activation with the levels of expression that we are seeing. And as well in platelet C, panel C, when we look at our platelets looking for the procoagulant surface marker, Nexin-5, we also do not see any kind of correlation there as well, indicating to us that perhaps the level of platelet activation is not necessarily correlated with the degree of exogenous protein expression of our luciferase reporter. One thing that is interesting, however, when we did look into this further for other platelet characteristics, was that we found a mild but statistically significant correlation between the amount of RNA in the platelets as well as how much protein is expressed in terms of, again, looking at our luciferase reporter. So when we looked at the immature platelet fraction as we measured through a Sysmex hematological analyzer, we noticed a slight negative correlation whereby platelets suggested to have a little more RNA content, less RNA content yielded higher levels of protein expression. And this is sort of corroborated by when we looked at the mean platelet volume, which again is an indicator of young versus old platelets, with larger volume indicative of younger platelets with more RNA content. We again saw a similar negative correlation whereby the platelets with a smaller total volume and again indicative of older platelets with less RNA seem to have a little bit higher translational capacity in terms of our luciferase reporter gene. And so while this is just a snapshot of some of the analyses we've been performing, we certainly have a lot more trying to analyze, and we are currently undergoing some proteomics and other transcriptomics as well to try to see if there's other markers that can correlate between the levels of platelet transgenic protein potential and the donor variability we're seeing. So certainly an ongoing process, and possibly would like to have the opportunity to give another update in the future. But thank you for listening, and happy to take any questions.
Sorry, is it more RNA or is it RNA stability? Is that something you guys are looking into? So it's not necessarily that there's more RNA, maybe it's just that it's more stable due to post-transcriptomic, like M6A mark changes or whatever. And
Right, that's actually a really good question. And so we haven't looked explicitly into the RNA stability in these platelets, so to say, and more so, again, just looking into the actual RNA content. Certainly, the stability of the RNA can play a factor, definitely in terms of how much protein is being expressed over time. We let these platelets translate to proteins just over a 24-hour cycle. And so in previous studies, when we looked at just how much RNA is remaining, we have noticed that a lot of the RNA we're delivering is still intact and still inside the platelet. But for this particular study across 81 donors, we did not look into that explicitly, but certainly a good thing to consider. Yeah.
It's more of a comment than a question. What you described about the less RNA is more effective. That's what we also see across the board. And we see that in skin, we see that in lungs, and I know it from other tissues as well. Less is more effective, and it goes down if you. So I don't know why. It's not necessarily viability or whatever, but this is very much in line with what we see in many others as well.
Right. Yeah, and certainly we're hoping that when we go deeper into our proteomics and transcriptomics analyses, for example, we might be able to shed a little bit more light in terms of what makes these different subpopulations a little different than the rest of them. Yeah.
Thank you.
Yeah.
Any other questions for Jerry? Jerry, thank you.
Yeah. Thank you.